Good afternoon, ladies and gentlemen. First of all, I would like to thank to the organizers for the opportunity to present our work today. My name is Agata Lange and I am affiliated with the Department of Nanobiotechnology, Institute of Biology at Warsaw University of Life Sciences in Poland. Today I will present our research entitled When Graphene Oxide Meets Silver Nanoparticles, Unlocking a New Era of Antibacterial Textile Coatings. The work I am going to discuss is covered by patents granted by the Patent Office in the Republic of Poland. In this presentation, I will demonstrate how the combination of graphene oxide and silver nanoparticles can be used to functionalize natural textile materials and create coatings with enhanced antibacterial and antibiofilm properties. I will cover both the physicochemical aspects of the developed nanocomposite, including its characterization and incorporation into the textile materials, as well as the biological and microbiological aspect. I hope this presentation will provide insight into the potential of nanotechnology-based textile coatings for future biomedical and protective application. Thank you for your attention and let me begin. Before discussing the developed nanocomposite, it is important to highlight that both graphene oxide and silver nanoparticles possess unique properties that contribute to their antibacterial activity. Graphene oxide exhibits several characteristics that make it an attractive antimicrobial material. Its sharp edges can mechanically damage bacterial cell walls and membranes, while its ability to isolate microorganisms from the surrounding environment may interfere with nutrient exchange. In addition, graphene oxide provides a large surface area, making it an effective platform for immobilization of the other nanoparticles, such as silver nanoparticles, and it offers a high ease of functionalization, enabling the design of multifunctional materials. Silver nanoparticles, on the other hand, are among the most widely studied antimicrobial nanomaterials. They offer several advantages over conventional antibiotics, including a multi-target mode of action, high reactivity, and the ability to penetrate biofilm structures, which is really important. Furthermore, silver nanoparticles are widely used in biomedical and antibacterial applications. When graphene oxide and silver nanoparticles are combined into a single nanocomposite, as I showed here, their properties can complement each other, leading to synergistic effects that enhance antibacterial performance. Such nanocomposites have been reported to exhibit, for example, strong antibacterial activity, but also reduced biofilm formation and thickness, disruption in the extracellular polymeric matrix, or the platform from graphene oxide exhibit enhanced colloidal stability for the metal nanoparticles. These advantages make graphene oxide and silver nanoparticle nanocomposite promising candidates for the development of next generation antibacterial coatings, including textile materials. Before discussing our result, it is important to address one of the major challenges facing modern medicine, antimicrobial resistance. Uh, it is often referred to as the silent pandemic of the 21st century and is recognized as one of the top 10 global public health threats facing humanity. The increasing prevalence of antibiotic resistant microorganisms has significantly reduced the effectiveness on conventional antibiotics and antimicrobial therapies, creating an urgent need for alternative strategies to prevent and control bacterial infections. One of the key factors contributing to antimicrobial resistance is the ability of microorganisms to create and to form biofilms. A bacterial biofilm is a three-dimensional community of microorganisms embedded within self-produced extracellular polymer matrix known as EPS. These protective structures shields bacteria cells from environmental stress, host immune responses, and antimicrobial agents, making biofilm associated bacteria up to 1,000 times more resistant to eradication than their free-living planktonic counterparts. In history, microbiology research focused primarily on planktonic bacteria, as these cells were easier to culture and investigate under laboratory conditions. As a result, biofilms were often overlooked despite their widespread occurrence in natural and clinical environments. Today, we know that biofilms can develop on both living and non-living surfaces, including skin, chronic wounds, medical devices, textiles, or everyday use objects. Due to their highly organized structures and complex life cycle, including attachment, maturation, dispersion stages, biofilm represent a significant challenge in infection control. Their presence is associated with numerous persistent and recurrent infections, particularly, as I said before, chronic wounds infections where biofilm formations delays. Healing promotes bacterial survival and reduces the effectiveness of antibiotic treatment. Therefore, the development of materials capable of preventing bacterial adhesion and disrupting biofilm formation is becoming increasingly important. This is where graphene oxide, silver nanoparticle, nanocomposites offer a particularly promising solution due to their combined antibacterial and antibiofilm properties. Considering the growing threat of antimicrobial resistance and the critical role of biofilm in persistent bacterial infection, the aim of this study was to develop natural textile materials functionalized with the graphene oxide and silver nanoparticles nanocomposite, exhibiting antibacterial and antibiofilm properties. The nanocomposite was incorporated into both cotton and silk fabrics, and the resulting materials were evaluated in terms of nanocomposite integration, physicochemical and mechanical performance, and antibacterial efficiency against representative bacterial pathogens. Briefly, the study began with the synthesis of graphene oxide and silver nanoparticle nanocomposites using an ultrasonic associated method. The obtained nanocomposite was then characterized by dynamic light scattering to measure the average size, then the zeta potential scanning transmission electron microscopy, and EDS analysis, and also its antibiofilm activity. Subsequently, nanocomposite was applied to the two natural textile materials, cotton and silk. The functionalized fabrics were assessed for nanocomposite incorporation efficiency, mechanically performance through peel adhesion and air permeability tests, and antibacterial activity according to the ISO 20645 against Staphylococcus aureus and Pseudomonas aeruginosa. With the methodology established, let me now move on to the results and examine the performance of the developed nanocomposite and the textiles. Dynamic light scattering showed an average particle size of approximately 214 nanometers and a zeta potential of minus 41.7 millivolts, indicating a good colloidal stability. The particle size obtained by DLS was consistent with scanning transmission electron microscopy, and the results are here, which confirmed a successful formation of the nanocomposite, revealing graphene oxide sheets decorated with silver nanoparticles. And here are the silver nanoparticles and the graphene oxide sheets. These results demonstrate the successful synthesis of a stable graphene oxide and silver nanoparticles nanocomposite. The antibiofilm activity of the nanocomposite was evaluated using confocal laser scanning microscopy and scanning electron microscopy. For confocal microscopy, biofilms were stained with DAPI and with Cyto9 to minimize non-specific bonding and staining and enable accurate visualization of biofilm architecture and thickness. The results demonstrated a great reduction in biofilm thickness following exposure to nanocomposites compared to untreated control. Here are the control sample for Staphylococcus aureus and Pseudomonas aeruginosa, and here are the groups with the nanocomposite. These findings were further confirmed with scanning electron microscopy observation. In the control sample, bacteria formed a dense three-dimensional biofilm structure with extensive cell-to-cell interactions in case of those two bacteria. In contrast, in groups with nanocomposites, samples exhibited a dramatic reduction in biofilm coverage. The biofilm architecture was severely disrupted, and only isolated bacteria cells or small cell clusters, for example, here, were observed on the surface. Together with confocal and scanning electron microscopy analysis clearly demonstrate that the silver nanoparticles with graphene oxide, the nanocomposite, effectively inhibits biofilm development, disrupts mature biofilm structure, and significantly reduces bacterial colonization. Encouraged by the strong antibacterial and antibiofilm activity of the nanocomposite, we proceeded with textile functionalization. Following sterilization, cotton and silk fabrics were sonicated in the nanocomposite suspension, enabling effective deposition of the nanocomposite coating onto the textile fabrics. Importantly, sonication did not adversely affect the structure of either textile material. After coating, the samples were dry and then analyzed to assess the uniformity of the nanocomposite coverage and to confirm the successful incorporation of graphene oxide, silver nanoparticle nanocomposites into the textile fabrics. Here, the micrographs reveals graphene oxide sheets spanning between textile fibers, as highlighted here by those red arrows. In some areas, the nanocomposite also wrapped around individual fibers, confirming successful incorporation and the good interaction of the nanocomposite with both cotton and silk material. Following the successful incorporation of the nanocomposite into the textile structure, an important question emerged. How stable would these functionalized materials would be during practical use? And to answer the question whether the nanocomposite could detach from the textile structure, we perform a peel adhesion test. In this experiment, the functionalized textile sample was pressed against the carbon tape, as we can see here in this picture. The use force was around 1 Newton, and subsequently the sample was peeled away. This procedure was designed to simulate mechanical stress and evaluate stability of the nanocomposite coating on the textile fibers. Here are also not only the picture of the peeling, but in some case the scheme of the procedure. As illustrated in the pictures, only one detached graphene oxide sheet was observed on the silk sample after the peel test, whereas no nanocomposite detachment was detected on cotton. Here are the two pictures. To answer the second question regarding the potential use of these materials, for example, as a protective face mask, we simulated breathing conditions using an artificial airflow model. The textiles were exposed to an airflow pressure. It was around 15 pascals, and the cycles 1200. It corresponds to around 1 hour of physiological breathing. This enables us to assess the stability of the nanocomposite coating and the fabrics under realistic conditions. Similar to the peel adhesion test, the airflow experiment demonstrated excellent retention of the nanocomposite within the textile structure. In the case of silk, only a single graphene sheet was detected on the surface of the upper filter, because here were the double filter. The use of a double filter system allowed us to assess the potential release and transport the nanocomposite fragment through the airflow pathway. The absence of the graphene oxide sheets on the lower filter for both textile materials indicates that the nanocomposite may remain strongly associated with their fabrics. To evaluate the antibacterial properties of the functionalized textiles, we performed one more test according to ISO 20645, a standard method specifically designed for antibacterial assessment of textile materials. In brief, the textile samples were completely dried and then placed on agar plates, inoculated with bacterial suspensions. Following incubation, bacterial growth was evaluated both around the sample, around the sample, by measuring the inhibition zone, and directly beneath the sample. Based on the criteria defined in the ISO protocol, a performance rating was assigned to each material. Our result demonstrated that cotton and silk with nanocomposite achieved a rating satisfactory against both Staphylococcus aureus and Pseudomonas aeruginosa. In summary, we successfully developed nanocomposites coated cotton and silk fabrics with antibacterial and antibiofilm properties. The nanocomposite was stably incorporated into the textile structure, remaining attached even under mechanical stress and simulated breathing conditions. Moreover, both materials demonstrated satisfactory antibacterial activity against Staphylococcus aureus and Pseudomonas aeruginosa, highlighting their potential for biomedical and protective textile application. And thank you for your attention.